microtubules are organized from this microtubule organizing center. So the minus ends sit in there, and they start at this special isoform of tubulin, gamma tubulin. That's very stable, and that really helps prevent the minus cap from the minus end from shortening. So they cap it. So now the dynamics really occur only on the plus end that's growing and undergoing those catastrophes. Let's see, what else do we want to say about this? So microtubule organizing centers, very important. They often sit close to the nucleus, and then the microtubule skeleton radiates out from there. In neurons, we see them right at the beginning of the axon, and so you will have, as long as the axon is long, microtubules extending all the way down so that the vesicles with neurotransmitters, whatever is needed at the very end of that axon, can be transported all the way over there. If we are dividing ourselves, we need to break apart our DNA, and then so we have those two spindle poles that are each a little microtubule organizing center from which the tubules are growing, attaching to the DNA, and then are able to pull it apart. So those were the things I wanted to show you guys regarding microtubules. We can see these as sort of a unidirectional, directional highway system across which motor proteins are sitting, and they could be driving things from the organizing center out to the periphery, and there's other motor proteins that are bringing stuff back to the center. And we'll talk about the motor proteins in a little bit, but first quickly talk about the intermediate filaments. These were the ones not made up of individual smaller proteins that then polymerize. These were encoded as one big strand. And we said that they were supporting the nucleus, so the inside of the nucleus. We can see that meshwork right here on the inside of the nucleus. And this might seem trivial, but this has to get broken down each time the nucleus, the DNA divides, and the nucleus has to split in two, and then it's reassembled. And if something is wrong, we can actually hit some very serious side effects. And one of those is a disease called progeria. So this is very rare. I think, did I write that in there? I think there's maybe been 180 cases since it was first described. So really very, very rare. It's a de novo mutation as these babies are developing. And what happens is initially we don't really see much, but within the first 2 years of life we start to see the symptoms already. And that is that they're not growing as fast. They really look like older people. They have a very characteristic bird-like face. And all of this results in them living much shorter, so they just really quickly age, and so they die of problems that we normally associate with people of old age. And this has to do with a single mutation in one of those intermediate filaments. A very common one, lamin A, has a single mutation that creates a shorter form of this lamin. So now it's no longer a long filament that helps provide structural integrity to the nucleus. And because here is a normal nucleus, here is one from a patient with progeria, and you can see that the structure is different. What they've stained for here is two different proteins, lamin A and lamin B. And you can see that one of those is not included in these little things that seem to be budding off of the nucleus. And so having a mutation in lamin A, I guess, doesn't make it interact with lamin B very well. And so now we get these areas where lamin B is excluded, and they bud off from the nucleus. The final result of this is that the nuclei undergo much faster cell division, or promoting cell division. And so these children are hitting their Hayflick limit much sooner in life, because the cells go through their maximum number of cell divisions. And this is what happens as we age. Our cell divisions slow down, there's no tissue replenishment, and this is called aging. That's why your skin wrinkles and your stuff doesn't work as well anymore. And so this just happens super fast in these kids because of this one protein mutation. Good. I'm kind of rushing through this because I feel like we spent quite a lot of time on the microtubules, and we have some other stuff to cover. So, molecular motors, which will be kind of our introduction for the muscle lecture. So myosins are examples of molecular motors. These are proteins that are actively moving along the cytoskeleton. Myosin is one that interacts with actin, and it's a plus end directed molecule, so that means the myosin protein is moving towards the plus end of the actin molecule. And so myosin is this really long stretch, and then there's a different domain of the myosin, which we call the myosin head, and this is actually where it connects to actin. So all of this sits kind of alongside actin, this interacts with the actin, and then you see here drawn some light chains. Those are regulatory subunits. We're not really going to talk about those, but they're often forgotten, so it's nice that they're actually included in this picture. So this is not one protein, right? We see here two green myosin heavy chains wrapped around each other, and then we have two actually myosin light chains, two different ones also attached. And this is what we often think of as a myosin, but it's actually a couple of myosin proteins together. And so this protein is going to crawl along actin by moving this head sequentially all the time. And that makes it then crawl along the actin. And here they've actually done a nice experiment. So this is a glass slide here. They've put on some myosins and they've stuck those tails to the glass, but the heads are free to move. And then they've fed it some actin microfilaments. And what you'll see is that those actin microfilaments are moving in the direction of their minus end as they hook up to the myosins that are stuck. Because the myosins are stuck in this case, so they can't move, but they're still trying to crawl along the actin, and the actin, therefore, which is free to move, is moving along the glass slide. And so here they've taken a black and white image where the actin is stained, and now they've artificially added a red color to this strand and a green color to this strand. And then they've followed what happens to these actin molecules over time, taking a picture every so many seconds or so many minutes. We call that a time-lapse video. You just take pictures, and in the end, when you put those all together, you get a nice movie of what's happening. And so you can see that initially this actin molecule sits here, but then it's actually moving up and up and up, and it will disappear out of your view over time. So if the actin molecule is moving towards its minus end, what does that mean for the direction that the myosin is trying to crawl? Is it trying to crawl to the minus end or to the plus end? Plus? It's trying to crawl to the plus end, right? So it is trying to move away from here to here to here to here towards the plus end, but because it is stuck, it's actually moving the actin filament in its minus direction. Really cool stuff. Fun experiments, I guess, to go ahead and do. Movement takes energy, and so myosins use ATP to generate this movement. And so they will take up ATP, they hydrolyze it, and the energy that is released from that is used to do these motions, and we'll talk in much greater detail about that in the muscle lecture. They move in a specific direction, and so we said now that the myosins are plus end directed. But there's other motor proteins that are minus end directed because we need to be able to transport things in our cells going both ways, right? Towards the periphery and from the periphery. They can carry different things or cargoes. They can be organelles, small vesicles, other proteins. You can imagine having this little motor protein carry an entire mitochondria along the side of the skeleton. So these guys are really, really good, right? Imagine the size difference. And so they're using energy to move stuff along. Here's two examples of those transport proteins, the motor proteins that use microtubules. So we just talked about myosin that uses actin. These guys are traveling on the microtubules made of tubulin. So dynein and kinesin are moving things in opposite directions. So if you start them at the same spot and you let them go, they're going to be walking away from each other or trying to walk towards each other. Both of them are dimers with ATP binding heads and a single tail. And so they can transport one thing, and they use these kind of like their feet. We'll see some movies where we see them move and stepping along the microtubule. So kinesins generally go towards the plus end, so they're similar to myosin on actin in that respect, and the dyneins are moving towards the minus end. So kinesins will take stuff out to the periphery of the cell, and then the dyneins are taking things back closer towards the center of the cell. Yes?
So on microtubules, we have proteins that move things in both directions. But on the filaments, do we also have something that'll move, a protein that'll move things in the minus direction, or is it just myosin that moves it in both?
I think most famously myosin, I imagine there must be things transporting it the other way. But myosin, because it's involved in muscle contraction, is usually the one that we highlight.
Thank you.
Different motor proteins transport different cargoes. It all depends on what you can bind, but they go by the same principle. So we'll have many kinesins. They can transport mitochondria, vesicle proteins, you name it. They're going to go in different directions. So if they're walking along one microtubule, they don't usually bump into each other. There's a lot of space for everybody to move around. And the tail, because that interacts with the cargo, the kind of tail that you have determines the kind of cargo that you can carry. All right, so here we see some examples of the stuff that gets carried around in the cell. So we have our microtubule organizing center here, where all the minus ends of the microtubules are inserted. And so we'll have kinesins that are trying to take stuff out, whereas the dyneins are trying to take stuff back in. So stuff coming in from the cell periphery, those would be things like endosomes, where we've taken stuff in from the plasma membrane and it needs to be transported towards a lysosome. Let's see, what else? Secretory vesicles are coming off of the ER and the Golgi, and they need to be going towards the cell membrane, otherwise the stuff will not be secreted. So this is pretty straightforward. I'm not sure I agree entirely with this example here. The ER does usually hang out close to the cell center. But apparently sometimes ER gets transported out. Alright, this movie you guys are going to have to watch all by yourself, but this one is so cute that I want to go ahead and watch it. Oh, and the other thing that's really nice about it is that this is, what's that mean? This is made in by a lab in the Netherlands. So the images you'll see will be of the town Utrecht. Utrecht. No, yesterday he actually made sound.
It's muted.
Oh, did I turn it off? How about that?
Our brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon. This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axon. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place. To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are two kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now. Fate strikes again. The other companion, Dynin, wakes up, and he can only walk in the opposite direction of John, resulting in a tug of war. But there can be only one. Along the axon in which John travels, there are places called synapses. Here, the axon connects to receivers of the other cells. Regulating proteins call the shots here. This traffic police makes sure that all passing traffic gets to the right destination. If John's cargo is needed in this synapse, he will be stopped and myosins take over his load. But today, John's cargo is safe. But what he does not know, that his road is under construction, just a few blocks away. In our nerve cells, the cytoskeleton is changing constantly. Roads are built, but are also broken down. Facing this kind of obstruction, John has to find a detour. John isn't the only motor protein on the road. There are many more. Our nerve cells need a smooth traffic flow in order to perform well. A traffic jam due to problems during the journey may ultimately result in brain disease. Understanding the challenges John faces could improve treatment or prevention. Finally, John arrives at his destination. He has fulfilled his destiny.
It's over here.
But several other Johns are just getting started.
That was cute.
I'm gonna go to the movie theater and record the movie.
watched this twice already. Yes, anybody wants to comment on the movie? Or shall we just move on to Muscle? Yes! So John was the Kinesin, right? Pulling it out to the plus end. That was Damien, the little guy whose slippers stay on the road. He's pulling it on the microtubule, but he tries to go the other way. And so the other movie that we didn't look at, they animate the motor proteins very, very similarly. So somehow they must know that this is how those guys walk, with that kind of floppy gait with the puffy slippers. Yeah, so those were images from Utrecht, one of our main cities. And as you can see, just like a main city here, I spent some time in Boston when they were doing the big still. It was awful. Always trying to find a new way, one-way streets, narrow streets. And Amsterdam is even worse because there they put up, for some reason, they put up the signs that say one-way street, like up in the second story. So you're driving and you don't know. So usually when we take our driving lessons, we always end up going to Amsterdam at least once. If you're not from Amsterdam like me, it was terrifying. Alright, so then we'll move on to muscle. I guess we'll have to extend this into tomorrow's lecture and then also try to work our way through the hormones tomorrow. So we'll talk about muscle structure and we'll really quickly delve into the proteins that are actually involved in contraction of the muscle. And I'm asking you guys to know a lot about this, but before we go all the way to the proteins, let's make sure that we know where we all are, right? So skeletal muscle attaches to your skeleton. So here's a picture of the muscle. Here's a picture of the bones. The muscles attach to the bones, and then by contracting, becoming shorter, they move the bones, allowing for movement. They don't attach straight up to the bones. There's this tendon here, and within the tendon, this is it's strong, but it's not as hard as bone, and it's a little more flexible, but not as good as the muscle. So it's a really perfect transition between bone and muscle, and even within the tendon, the properties, it's much stiffer close to the bone, and it's much more flexible closer to the muscle. And so this bundle of muscle is wrapped in this fascia, this collagen-rich sheet that will terminate in the tendon. And then if you go in, we see here these bundles of muscle cells that are wrapped up in yet other layers of collagen and stuff that you can excuse me, probably also see when you buy a nice piece of steak. I always look for steaks with some fat in it. That's somewhat different, but that's also stored in between here just to have some extra energy. But here we have a bundle of muscle cells that we see on cross-section, and a muscle cell is referred to as a muscle fiber. This here is an enlargement of a single muscle cell, and we're looking at some of those bundles of proteins that are going to be doing the contractions. And the funny thing that you see is there's a nucleus here, but there's another one right here, and this is the same cell. And that's because as muscle develops, the cells first multiply, but then they fuse. So it's not that they forget to divide, they fuse after they've grown a lot. And so this means that we have this really, really long cell because muscle fibers can extend the entire length of your muscle. And to make up for having to cover such a long distance from the nucleus to whatever site in the muscle cell you would be at, they've just decided to work together and actually be a single cell made up of multiple cells, so with multiple nuclei, allowing for proteins to be produced much more locally. It's much more efficient. But they just have omitted the membranes that would normally separate them. So in the muscles, there's all these bundles of contractile proteins, and if you take one of those bundles out, we call those myofibrils, then we're getting close to where we're going to be focusing today's, or the rest of today's lecture. Right, so we have myofibrils, those are bundles of contractile proteins. A bunch of those bundles are stored in a muscle fiber, which is basically a muscle cell. And then a bunch of those muscle cells are stored here together. And then a bunch of those bundles finally start getting us back towards the whole muscle. But it's really layer upon layer and initially very confusing. For the lecture today, we'll focus here because this is where the actual action is taking place. And you can already see that there's different types of proteins here, this purple fatter one and then this thinner blue one. These are part of the slides that I uploaded today. They weren't there earlier, so I don't know if you took them down earlier. Then you won't have this slide and the next one. But I thought it was more clarifying to have them present. So, hey. The picture on the next slide shows you an area in between this sort of blue line going down and this blue line going down. So we're really looking at a very small area of muscle. Again, we have the purple, in this case pink, thicker proteins, thick filaments. Those are made of myosin. And then we have these blue, thinner filaments made of actin. So the name filament now is confusing to us, right? Because it's not made up of multiple small myosins coming together to make a long chain, but that's what they were calling it in muscle. So this bundle of all these thick and thin filaments together organized in a very specific way, that's the myofibril. If you look at it on cross section, you can see the thick and the thin filaments there. This is an enlarged picture of an electron microscope image. And if you draw this, you can see that each thick filament is surrounded by 6 thins, and each thin is surrounded by 3 thicks. So very, very organized. And we'll be talking about this thing cross-bridge cycling because that's what's needed. The myosin and the actin need to physically touch each other in order for movement to take place. And so those are the cross bridges. A piece of the myosin sticking out and it's going to touch the thin filament made of actin. So now if we slice it here and then all the way across and take an electron micrograph, we can see again this area of the blue line coming down. This was the blue line in the pictures before. This was purple initially, so these are your thick filaments. This is all myosin that you see here. And then those thin filaments come off from the perpendicular layer, and they stick in between these thick filaments. Right? They stick in between these thick filaments. So there's an area where myosin is sitting, there's an area where we see actin all by itself, and then there's an area where the two of them overlap. And because it's so easily recognizable and it looks so striking, this is why we call skeletal muscles striated, stripy looking, because this pattern repeats all the time, light, dark, light, dark. And so this dark area, that's where myosin is sitting. That's a big protein, it's electron dense, so we can see it as a darker area in our electron microscope picture. Actin and the other proteins that it associates with is lighter electron dense, so it gives you a lighter color, and the actin is also present here. Now you'll see here indicated H zone here. That's the area where myosin is sitting by itself. So the actin sticks in a good way, but not all the way. And then in the very middle, there's again this darker stripe where the myosins are connected to each other, and you call that the M line. And this big one where the actins are attaching is the Z, for zwischen, the German word for in between. And so in between two Z lines, the proteins that are present there make up the sarcomere. And so the sarcomere is the smallest unit of muscle that has everything that you need to make a muscle contract and become shorter. It has the actins, it has the myosins, it has a place where they overlap and where the myosin can crawl over the actin to bring those two Z lines closer together. Good. That for orientation. Questions on this, or we'll just move straight on to how this myosin then looks in the sarcomere. Actually, it's kind of dark still, right, for the video. So myosin, like we said, is this large molecule, the heavy chain. And then each heavy chain is interacting with two different light chains that we're not going to really worry about for now, but they both have a unique sequence, unique function. We'll just put them there, and then I guess in 110C you'll learn more about those. So this combo together looks like this, and then it finds another myosin, and their tails wrap themselves around each other, they dimerize, and so now we have a half functional myosin. Two of those units come together end to end. So this would be the M line, right? The very middle of the sarcomere. And these guys now are going to sit in between bundles of actin. That's what we see here, these lighter yellowish proteins. And the cross bridge then is the connection between the myosin tail and the actin filament made up by the myosin head. So that's the cross bridge that's forming to actually make the connection. So here the same structures are indicated as we saw before in the electron micrograph. The whole thing between the two Z lines is called the sarcomere. We have actins coming in from either end, and these actins are inserted with their plus end into the Z line. So the plus end is not growing. And I imagine that the minus end is also somehow capped and protected, that this is a stable actin that's not growing or shrinking. Let's see, the myosins sit in between, and again, the area where actin isn't reaching is called the H zone, and then this A band here is where we find our protein myosin, and the I band is where we do not find our protein myosin. Right, so the area of overlap is within the A band. So far we've looked at the thin filament as being a thing made up of just actin, but that is actually not true. That too is made up of several different proteins. So we have our double helix of alpha, of actin, and if you can see that there's this thing that wraps itself around it, stabilizing this microfilament, and that's trypomyosin. And what it also does is it's covering up this little area here where myosin likes to bind to actin. So if this is wrapped around it, myosin is unable to bind. And in order for us to have a way to regulate whether or not myosin can bind, that's why we have those troponins. Troponin T interacts with the trypomyosin. Troponin C interacts with calcium, that's why it's called C. And troponin I brings it all together and makes this complex inhibitory unless we have calcium present. And then these troponins, the T and I, that's actually what people measure if it is suspected that you had a heart attack. Because if you have heart damage, muscle proteins are released in your blood. And so they actually measure these proteins, troponin T and troponin I. And one of our colleagues in NPB, Dr. Gomes, somehow at some point was involved in developing this assay, which I thought was pretty cool. And it's used a lot, so it's a very reliable test for muscle damage. So, we said if calcium is not present, this process of actin and myosin interacting is inhibited by the trypomyosin. So how does this change when calcium shows up in the cell? When calcium shows up, it binds to troponin C. that then ends up pulling away the entire complex from those interaction sites. And so now instead of having closed myosin binding sites, the triple myosin is removed, and now all of a sudden, the favorite place for myosin to interact with actin is wide open. So now myosin can come in and bind, and we have a cross bridge established. So then the question becomes, well, how do we get calcium into the muscle cell, right? If this is what regulates that we can make muscle contraction happen, how do we get calcium into the muscle cell? This might look very familiar because here we have a nerve terminal. This is a motor neuron. It has acetylcholine as its neurotransmitter. So when this receives an action potential, the electrical signal, it changes that to a chemical signal and releases acetylcholine in this cleft here. But instead of having a neuron on the other end, we have a muscle cell on the other end. So we have here the muscle membrane. And there's these deep invaginations in skeletal muscle membranes that are really useful for speeding up the rate of information transfer from the neuron to actually making muscle contraction happen. And on the membrane of this muscle cell, we have acetylcholine receptors that are sodium channels, right? And you guys will, of course, all remember from Dr. Bautista's lectures that if you open a sodium channel, sodium is going to go in or out of the cell. In. Excellent. And so sodium goes in, the membrane depolarizes, and we are generating an action potential. So the membrane becomes more positive. The action potential travels away from this site where the nerve is attaching, and it goes down into this invagination, this dip of the plasma membrane. We call that the T tubule. And what sits down below here is this DHP, dihydropyridine receptor. This guy is sensitive to voltages, and if the voltage changes, then it's going to change its shape and pull open this door, which is made up of another receptor. This other receptor, ryanodine receptor, sits in the sarcoplasmic reticulum, not the one with all the ribosomes, but the one that stores calcium. So if the action potential comes in here, this changes shape, pulls open the door of the ryanodine molecule, and calcium is flooding out and flooding all these actins and myosins. So now the process that we just described can take place, and the myosin and the actin can interact. So, put it here. We have a quicker question just in time before end of lecture.
You read the whole thing?